We're delighted today to have the NIEHS Distinguished Lecture from Lindsey Criswell, the Director of the National Institute of Arthritis and Musculoskeletal and Skin Diseases at the National Institutes of Health, and the head of the Genomics and Autoimmune Rheumatic Disease section in the Medical Genetics branch of the National Human Genome Research Institute. Lindsey is a rheumatologist and human genetic epidemiologist who has played a major role in identification of genes and environmental factors that contribute to risk and outcome of autoimmune rheumatic diseases. She earned her medical doctorate at UCSF, her MPH degree at University of California, Berkeley School of Public Health, and her Doctor of Science in Genetic Epidemiology at the Netherlands Institute of Health Science. She became a rheumatologist at UCSF, rising through the ranks to professor and chief of the Division of Rheumatology, and later serving as the Vice Chancellor of Research at the University of California at San Francisco. She has had substantial training and experience in epidemiologic and statistical methods for identifying genes that contribute to complex human disorders. She's contributed to genome-wide association studies of systemic lupus erythematosus, rheumatoid arthritis, and Sjogren's disease, resulting in the identification of many genes that contribute to these disorders, as well as identifying many pathways involved in these diseases and important therapeutic targets. In addition to leading projects that have sought to refine genotype and phenotype associations for specific manifestations of autoimmune diseases, she's also sought to determine epigenetic factors, including DNA methylation status and contributions to the risk and outcome of these diseases. She's had experience recruiting and enrolling autoimmune disease patients for participation in consortium studies and led to the development of national and international consortia that have been very important in the field. As Vice Chancellor of Research at UCSF, she was also responsible for directing efforts to advance innovative, collaborative, and interdisciplinary biomedical research across professional schools at UCSF and across the graduate programs. She also served as the program director for the NIAMS P30 Precision Medicine and Rheumatology Center and was co-PI for the Clinical and Translational Science Institute at UCSF. She has mentored a large number of medical and graduate students and postdoctoral fellows. She's received a number of distinguished awards, including as a member of the Association of American Physicians. She has a very strong interest in the environment and autoimmune disease, and so we're extremely delighted for her visit here at NIEHS. And we welcome her lecture on the epigenetic contribution to systemic lupus erythematosus. Thank you so much, Lindsey, for joining us.
Wow. I think that was the longest introduction I've ever received, and I have never been taken better care of than by Lisa. So it is such a privilege and a pleasure to be here. So thank you so much. And I hope you enjoy what I have to share with you. My comments won't take me too long, so I hope to leave plenty of time for questions at the end. And I have no disclosures to share. So I know most of you are not rheumatologists, so you may not be familiar with systemic lupus erythematosus, or SLE, as you'll see, I'll refer to it. But it's considered the prototypic systemic autoimmune disease. It is characterized by striking clinical heterogeneity, which will be a major focus throughout my comments today. Women are at dramatically increased risk of developing the disease, and in the case of lupus, individuals of non-European ancestry are more likely to get the disease, and they have more severe disease, something that we're still trying to understand. The disease is associated with substantial morbidity and mortality. The etiology is complex, genetics, environment, etc. There is still no pathognomonic test for diagnosis, and this is a chronic illness with no cure, so very compelling. But genetic and other etiologic studies of lupus are complicated by that striking phenotypic heterogeneity, which I've illustrated on this slide, which shows just some of the many manifestations that individuals might present. They include various types of skin disease, some of which have very significant consequences, Joint involvement with arthritis is common. The central nervous system can be involved in some patients. Kidney disease, as reflected by this pathology slide of lupus nephritis, is one of the most common but worrisome complications of the disease. It's associated with very substantial morbidity and mortality in lupus. But lupus is also associated with a wide range of autoantibodies that have differential associations with disease manifestations. There's also increasing evidence that specific autoantibody profiles may be more closely linked to genetic or epigenetic factors than the overall disease status. This slide illustrates some of those autoantibodies. For example, anti-double-stranded DNA antibodies, which I'll refer to on several occasions, is associated with renal disease, quite specific for lupus. Anti-cardiolipin antibodies, on the other hand, are associated with various thrombotic complications of the disease, etc. Now, I mentioned we have no pathognomonic test that allows us to say whether a person does or doesn't have lupus. What we have instead are what we call classification criteria. And if you consider these various manifestations, you can understand what I mean by the tremendous heterogeneity that's encompassed. The way these criteria work is that an individual who meets 4 out of the 11, any 4, at any point in time, can be classified as having lupus. But that allows for a tremendous amount of variation. That is a challenge for us clinically and in terms of our efforts to understand this disease. Just one brief slide that tries to capture the strength of the overall genetic contribution to lupus relative to some other autoimmune diseases. This slide summarizes what we refer to as lambda sub S, the S representing sibling, the lambda sub S measure of the overall genetic contribution to the disease. It's simply the risk of the disease among siblings of an infected individuals over the background population frequency. And although we don't have precise figures for these, if you look at lupus compared to these other autoimmune diseases, it conveys the fact that overall, the genetic contribution is relatively strong. And as many of you are aware, progress defining the genetic contribution to lupus was painfully slow until about 2007, 2008, when it became possible to perform genome-wide association studies in a cost-efficient manner. And starting at that point, there has been an absolute implosion of knowledge about genetic variants that contribute to the disease. I'll show you this meta-analysis performed published in 2015. This was a meta-analysis of 5,700 cases and 13,000 control individuals, all of European ancestry. And this study identified 63 risk signals with unequivocal statistical evidence at 52 distinct loci. Now, that very high peak on chromosome 6, which is actually truncated on this slide because it would go all the way off the top of the slide there, that corresponds to the HLA region on chromosome 6, a very strong risk factor, or that region encodes for very strong risk for lupus and other autoimmune diseases, at least in individuals of European ancestry. Now, we were intrigued with the extent to which the emerging GWA data in lupus highlighted the importance of this particular pathway that I'm referring to as the toll-like receptor type 1 interferon signaling pathway. Virtually every component of this pathway has now been implicated in GWA or other studies of the disease. And this is not to say that this explains the disease entirely, absolutely not, but the genetic data really reinforced some of the theories, hypotheses, and other data that was emerging about contributors to lupus. Now, one more Manhattan plot from a very recent GWA study in lupus. In this case, in a population including over 20,000 individuals from East Asian ancestry. And in this study alone, now we identify 113 risk loci. All of those peaks in pink were novel to this study. So we're continuing to learn more about the genetic contribution to the disease, even with the GWA approach. Now, currently, if you were to look in the NHGRI GWAS catalog, you will see that there are 183 risk loci, significant associations for lupus at this time. So really exciting progress. Now, a major focus of my lab has been on understanding the contribution of genetics and epigenetic factors to disease heterogeneity that I highlighted in the beginning. This focus of my lab was influenced by the results of this very early study we performed, actually before the first GWAS studies of autoimmune disease, when we identified a variant of STAT4 as the explanatory factor for a chromosome, a linkage peak on chromosome 2q in rheumatoid arthritis individuals. Now, at that point in time, I had been working very hard to create a research resource for lupus at my home institution, so we were able to quickly look at that variant in our lupus collection to see whether it was also associated with the disease. Now, not only did we determine that actually the association with lupus was stronger than the association with rheumatoid arthritis, but when we looked carefully at the phenotype data that we had painstakingly collected for all of these individuals, we found that the strength of the association, as reflected by the odds ratio there on the right side of the slide, varied widely depending upon the specific disease features that were present. So among individuals with what we consider mild disease features, oral ulcers, photosensitivity, the odds ratios were about 1.5, still impressive for a complex disease, but odds ratios exceeding 2 for individuals with severe nephritis. I had hypothesized that maybe arthritis in lupus was going to be the strongest association here, but actually arthritis, in spite of the kind of similarity to rheumatoid arthritis, did not stand out at all in this analysis. So inspired by those results, we then subsequently sought to elucidate the heterogeneity of genetic associations with emerging risk loci from GWA and other studies. Shown here are the results of our analysis of 2 of the initial GWA studies in lupus, which included about 1,700 cases and 4,800 healthy controls, European ancestry, in which we stratified the results by double-stranded DNA autoantibody status. So you have the Manhattan plot on above, just those cases that produce these autoantibodies, the Manhattan plot below, those who did not produce these autoantibodies. And as you can see, now these subgroups were equally sized, so we're not talking about a power difference here. The genetic architecture of these subsets that are classified all together as having lupus, quite distinct. For this study, we performed a meta-analysis of 3 genome-wide association studies in which we compared women with lupus and lupus nephritis to women with lupus who had not developed nephritis as part of their disease, so a case-only analysis. And several results were notable here. One was the relatively modest association with HLA. You can see it there in brown here, very modest relative to the other GWA, the Manhattan plots I showed you. Also, what was striking is that many of the top hits here had not been on our radar screens through case control studies. So we were identifying a new set of genes that contribute to the development of renal disease in lupus that were not necessarily contributing strongly to risk of the disease altogether. So that was a very important result, and in terms of how we can get insight to other factors that influence this heterogeneous disease. Now, it's been very clear from the result of twin studies, for example, where there's substantial discordance for disease among monozygotic twins, and other considerations that the environmental and epigenetic factors have a very important influence on the development of autoimmune diseases, lupus, and others. And I know that the Institute, NIHS, is very interested in this topic. However, it's been difficult to study the environmental contribution to the disease in a rigorous, systematic, and comprehensive way, as you all are aware. We've been interested in the epigenetic process of DNA methylation due to its important role in gene expression and the fact that it's affected by environmental factors. And indeed, increasingly, DNA methylation is recognized as an important biomarker for exposures. For those of you not familiar, DNA methylation is an epigenetic change that consists of the addition of a methyl group to DNA at a so-called CPG site, where you have a C followed by a G. And in general, more methylation leads to tighter packing of DNA and suppressed gene expression, and vice versa. And also importantly, different cell types have distinct methylation patterns that dictate what genes are available for transcription. I first became very interested in DNA methylation based on the results of this very small study that demonstrated striking differences in DNA methylation levels among a small set of monozygotic twins who were discordant for lupus. This study included 5 discordant twins, and 5 healthy control individuals. Now, at the time this study was done, they performed state-of-the-art methylation profiling, which consisted of the assessment of 800 CpG sites genome-wide. And as you can see from the heat map, overall, there were decreased levels of methylation status in certain regions compared to, among the cases, compared to the unaffected twins and controls. And when we looked at the genes that were significantly differentially methylated, many of them had logical roles for an autoimmune or immune disease such as lupus. So this was very exciting insight into the important role that DNA methylation, or how it might help us understand risk for the disease. Now, this work also increased rapidly. Just a year later, now it became possible to interrogate 28,000 methylation sites genome-wide. This is a slightly larger study of 12 cases and 12 controls, once again showing many differentially methylated regions, either hypomethylated or hypermethylated regions, distinguishing cases from controls. These are unrelated individuals. They also found some preliminary evidence of an association with disease activity, a very important concept in lupus that I'll talk about in a bit. Now, the largest one of these case control EWA studies, and EWA refers to epigenome-wide association studies, so these studies are often referred to as EWAS. The largest such study, published a few years ago now, was performed in a Swedish population and included about 550 cases, 590 controls. And just to show you how the technology has evolved, at this point in time, it was possible using the Illumina bead chip to interrogate 450,000 CpG sites genome-wide. Now, these investigators also performed genotyping, not genome-wide genotyping, but they characterized the subjects for the immunochip set of markers, which includes about 200,000 variants associated with immune traits. Once again, these investigators found, as we've seen, association of DNA methylation levels with disease status, but they were also able to show, because they had genetic data on these individuals, that a minority of these differentially methylated sites were under genetic control, and some of the genes associated within those relationships were lupus risk loci, suggesting that methylation may be mediating some of the genetic contribution to disease. Now, because we have been so focused on heterogeneity in the phenotype, we were very interested in determining whether DNA methylation data may help to explain disease heterogeneity beyond what we're learning from genetic studies. So shown on this slide are the results from an EWAS that we performed using that same Illumina array I just mentioned, and involving a comparison between women with lupus based on the presence versus absence of double-stranded DNA antibodies. So no controls in this analysis, just comparing lupus cases with and without that important disease manifestation. The results were notable for the relatively modest association with CpG sites in the MHC region, once again, very distinct from genetic studies, case control studies, and many top hits that had not been identified in genetic studies, suggesting that indeed DNA methylation data can add to what we're learning about lupus from genetic data. Now, similar to genome-wide association studies, there are many covariates and important confounders to be considered. So in this analysis, for example, we performed multivariable regression to account for differences in age, genetic ancestry, and cell type proportions. And we also restricted this analysis to women who are non-smokers to try to minimize the impact of some of those confounders. Now, similarly, in this analysis, we're comparing lupus cases with and without lupus nephritis. In this case, genome-wide profiling identified associations between lupus nephritis and differentially methylation of genes regulating tissue hypoxia and also type 1 interferon responses. And as I mentioned previously, dysregulation of type 1 interferons has been previously implicated in the pathogenesis of lupus. Now, we are currently characterizing a cohort, a multi-ethnic cohort of over 2,000 lupus cases with the latest and greatest Illumina array, the EPIC version 2.0 chip, which has over 925,000 CpG sites. So we're really excited to be able to continue our work in that much larger and more diverse sample set. Now, thanks to support from the CDC, I had the opportunity to contribute to the development of a unique resource for studying lupus called the CLUES, for California Lupus Epidemiology Study. The CDC has had a long-standing interest in lupus. This cohort was designed to be population-based, in this case in San Francisco County, and multi-ethnic, and also to follow patients longitudinally. Thus far, over 350 patients have been enrolled and deeply phenotyped, and I was able to secure funding from other sources to support biospecimen collection in this resource. We also recently contributed to the successful renewal of this cooperative agreement that will allow for ongoing enrollment of participants during disease flares, something that was challenging to capture with this study design, but that we're very interested in. Now, the deep phenotyping of the CLUES participants in conjunction with the molecular data that we're working to generate provides a unique resource to investigate disease heterogeneity in lupus. This study, led by Christina Lanata, who's now a staff clinician in my lab at NHGRI, leveraged genome-wide genetic and DNA methylation data applied to the CLUES cohort. We first stratified patients into clusters based on the American College of Rheumatology, the ACR criteria that I mentioned earlier, and the sub-criteria using an unsupervised clustering approach. That resulted in 3 clusters that were identified based on all of that data that differed in terms of severity, with one mild cluster shown both kind of in the table and the graph there in blue, and 2 severe clusters that had different combinations of manifestations shown in the green and the orange. Then analysis of genome-wide DNA methylation data identified 256 CPG sites that coincided with 124 genes that differed significantly across these clusters, including many that were in the type 1 interferon pathway. We were able to validate these results in an independent cohort that we had access to, and we performed cis-MEQTL analyses to identify cluster-associated CPGs that were under genetic control. And we also performed causal inference testing to identify cluster-associated CPGs that mediated genetic association with those clusters. And once again, here you can see down at the bottom the various covariates and confounders that we're accounting for in these analyses, which is critically important. We were then able to take those 56 CPG sites, map them into the corresponding genes, and then perform pathway analysis. And not surprisingly, the results showed significant enrichment of genes associated with type 1 interferon signaling that I've been mentioning, antiviral responses, something viral infection has been very interesting in lupus, and various inflammatory pathways. Now, as I mentioned, the CLUES cohort includes longitudinal data and biospecimen collection. So we've been interested in examining changes in DNA methylation during periods of high disease activity, that is flares, and low disease activity or remission. This is a very important clinical issue because patients with lupus flare, it's hard to predict when the flares are going to occur, what the flares look like, what is going to be required, how aggressive the treatment is going to be required to get the disease under control, et cetera. So this is a really important clinical need. Now, in this study, disease activity was measured with the SLE Disease Activity Index, or SLEDI, which is a validated instrument for measuring disease activity in lupus. And our initial analyses, looking at flare in lupus, included 59 patients who were sampled during periods of flare and about 3 months post-flare, as shown on this slide, with concurrent data collection, including medications, disease status, et cetera. And this slide summarizes very briefly our initial results examining longitudinal changes in methylation in relationship to disease activity. And these analyses were led by a fantastic postdoc with my NHGRI lab, Mary Horton, who joined us upon completing her PhD in epidemiology at UC Berkeley. So we found that differences in methylation patterns over time are associated with achievement of remission following a flare. The heat map on the slide shows that, for example, all patients in cluster 1, and the cluster assignments are in this row, all patients in cluster 1, which is on the left, achieved remission, every one of them, despite having similar disease activity levels at baseline and medications and demographic characteristics compared to the other clusters. Now, for this work, we used the Illumina EPIC version 1 array, which has over 800,000 CPG sites, so quite good genome coverage. And we adjusted for batch, age at which the diagnosis was made, the time interval between the visits, sex, medication use, and importantly, the cell type proportions. We were able to account for 12 leukocyte subtypes using the data. Now, DNA methylation occurs during gametogenesis and after fertilization. And after each cell division, methylation marks are maintained by the DNA methyltransferases. The majority of methylation marks are stable. However, DNA methylation can be influenced by a variety of environmental factors, as well as by oxidative stress. And as many members of your community, some of whom are here today, are very well aware, a variety of environmental factors have been implicated in lupus and other autoimmune diseases. However, there are significant limitations in the quality of much of the evidence, some of which is even more anecdotal in nature. Further, compared to genetic factors, the potential environmental exposures that might influence a disease is vast. This slide kind of nicely illustrates the strength of evidence for a variety of environmental factors that have been implicated in lupus. As I mentioned, NIAHS has had a long-standing interest in the environmental contribution to human autoimmune disease, as reflected by this report that followed an expert panel workshop back in 2012 and many publications since then. And it's really great to see Fred Miller here in the audience. So thank you for all of your work and the many people at NIAHS who have contributed to this very interesting area. Increasingly, as I've mentioned, DNA methylation changes have been associated with specific environmental exposures, suggesting that DNA methylation may serve as a useful biomarker of exposures. And this slide, which comes from a review that we published a few years ago, summarizes some of the exposures that have been associated with specific DNA methylation changes and that have also been implicated in lupus or related conditions. The concept of the exposome, which you are all very well familiar with, encompassing the totality of exposures across the life course, was introduced almost 20 years ago now. My lab has been interested in applying emerging technologies that enable more comprehensive and unbiased approaches for characterizing the exposome. So this past December, just a few months ago, we published the results of a proof of concept study in which we applied a non-targeted approach to detect circulating exogenous compounds in plasma using liquid chromatography quadrupole time of flight mass spectrometry, or LC-QTOF-MS. We used the CLUES cohort, and specifically we studied 285 lupus cases from that cohort and 103 healthy controls. And we observed increased abundance of acid pesticides and phthalate metabolites in the lupus cases compared to the controls. These chemicals have been previously implicated in lupus, making these results quite interesting. And I want to emphasize this. Although the cross-sectional design does not allow us to determine causality from this study, employment of this approach in population-based studies can generate hypotheses and inform future investigations of environmental factors contributing to lupus risk and severity. And I'm just including this one diagram from our paper that highlights the pipeline and some of the steps that are taken to generate this data and to try to analyze the data. And we've learned quite a lot in this initial exposomic study. In this case, the MS features that were present in at least 25% of all samples were selected for the association analysis. So that was about 2,700 features among the 71,000 that were identified. And then features were matched to potential chemicals using available databases. An association analysis of abundances of features with lupus status was performed, as I mentioned, adjusting for age and sex. And we also explored associations with lupus subphenotypes, with socioeconomic status measures that we've collected on all these individuals, and with current medication use. I felt very fortunate that since moving to the NIH, we've been able to establish a collaboration with the NIHS Metabolomics Core Facility and their integrative bioinformatics support group. And this slide summarizes some metabolomics data recently, as in a few weeks ago, generated in that core for an analysis in which we're comparing samples collected from patients during periods of high versus low disease activity. That's how the heat map is organized on the left side of this slide. So the heat map shows chemical features that were statistically significantly different between the high and low disease activity samples, with at least a 1.54-fold difference. We are very eager to extend these analyses to a larger sample set and to begin to integrate these data with other molecular data types such as genetic and DNA methylation data. Along these lines, I'm very excited about this new collaborative effort between the NIH Office of Autoimmune Disease Research, and in fact, for that new office, this was their inaugural project that they funded, so thanks to Rick and others who made that happen. That new office falls within the Office of Research on Women's Health, and NIAMS, NIHS, and other institutes are deeply interested in this topic and will be working moving forward to try to help this effort evolve. But just to tell you briefly about this new program, it's called EXACT, for exposome and autoimmune disease collaborating teams. And as the name implies, it seeks to develop a multi-site network that will adopt a team science approach to produce a systems-level understanding of the role of the exposome in autoimmune diseases. Now, lastly, I'd like to summarize some of our work in a related autoimmune disease, Sjogren's disease. Like lupus, Sjogren's is a systemic autoimmune disease. It's clinically heterogeneous. There's a striking female predominance. It overlaps with other autoimmune diseases, and it has a complex etiology. Importantly, there have been no approved therapeutics for Sjogren's disease, and the incidence and prevalence of this condition is increasing, as is strongly indicated by recent population-based studies in the UK and the US. The disease can affect almost any organ, but it's characterized primarily by inflammatory infiltration of the exocrine glands, resulting in ocular and oral dryness, which can be quite debilitating. Also, a notable complication of the disease is a dramatically increased risk of lymphoma. Shown at the bottom of the slide on the left is a study in which we documented involvement of TNF-AIP3 gene variants in risk of both Sjogren's and concomitant lymphoma development, but there's a lot more to learn about that relationship. Now, Sjogren's also shares a number of features with lupus. Indeed, in France, I spent a year there, Sjogren's disease is sometimes referred to as lupus des muqueuses, or mucosal lupus, recognizing that relationship. Thus, the availability of molecular data from these two related diseases provides a unique opportunity to investigate shared versus common risk factors and pathways. Now, like lupus, the classification criteria have been developed, and in this case, classification is based on the weighted sum of 5 items, which reflect the presence of lymphocytic infiltration of the labial salivary glands, right here in the lower lip, as shown in the histopathologic slide, presence of specific autoantibodies, and objective measures of ocular or oral dryness, as is captured in some of the images on this slide. Now, importantly, the study of Sjogren's syndrome lags far behind other autoimmune diseases despite this rising incidence. Our studies of Sjogren's disease leverage the unique Sjogren's International Collaborative Clinical Alliance, or SICA resource, that I helped develop along with colleagues at UCSF and around the world, including my co-PI Caroline Shabosky, who continues to lead this effort. We enrolled over 3,500 patients from 9 sites in 7 countries. We collected extensive phenotype data and biospecimens, 9 biospecimen types, including labial salivary gland tissue that was harvested from every single enrollee. And we ensured consistency in all study procedures through regular site visits to these US and international sites. Shown at the bottom is a picture of one of our sites, Aravind Eye Hospital in India, which is the world's largest eye hospital. And if I had more time, I'd tell you about that visit, which was pretty amazing. So using this cohort, we published the first multi-ethnic GWAS of Sjogren's disease, which revealed striking heterogeneity according to ancestry, something that we were not anticipating. The graph on the left shows association results for the MHC region among the European in blue and the Asian subgroups in red. Now we scaled these so that they're comparably in size. So this is not a power difference, but strikingly different. And then on the right, we have the Manhattan plots for the European subgroup up above and a meta-analysis of our Chinese and Japanese subgroups down below. So very, very different genetic contribution to the disease for these major ancestral groups. And this was in spite of matching on clinical features. So this was not explained by differences in the clinical manifestations. Now, as I mentioned, a unique feature of the SICA collection is the availability of the labial salivary gland tissue, which represents the target tissue for the disease. So we've recently published work showing that hypomethylation, now this is in the gland tissue, mediates genetic association with the MHC region genes in Sjogren's, and we've also used the methylation data to identify subgroups of Sjogren's that are not completely correlated or don't simply reflect our classification criteria. I wanted to show a picture of Lisa Bercelis on this slide because she's been such an important collaborator for all of our DNA methylation studies. She has a genomics lab at UC Berkeley. And I'm just going to show you one glimpse of our very recent data in which we're looking at an analysis of labial salivary gland DNA methylation data for about 1,000 SICA participants chosen from the 2 largest ancestral groups in that collection, which identifies 4 clusters that differ in terms of disease severity and proportions of estimated cell types. So patients in clusters 2 and 4, which are represented in the green and pink, show significantly more severe disease. Also, cases in clusters 2 and 4 exhibit a greater proportion of immune cells, particularly B cells, and lower proportion of epithelial cells compared to clusters 1 and 3. So, to summarize, tremendous progress defining the genetic contribution to lupus, but so much more work is needed in identifying the causal variants, defining the biological mechanisms, etc. It's certainly provided clues to etiologic pathways and, in some cases, therapeutic targets. There is dramatic specificity of genetic associations for disease manifestations, and importantly, as I've tried to emphasize today, epigenetic factors also contribute to lupus risk and disease phenotypes and provide a link to the environment, which has been so challenging to characterize. We're very excited about new methods that allow for an unbiased identification of relevant exposures, the exposome or metabolome, and importantly, integrative analyses are going to be needed for a full understanding of the biological pathways and the molecular targets for better therapies and better understanding of the disease. And ultimately, our vision is that this work can help to delineate a molecular taxonomy of disease that is badly needed given limitations of the current classification criteria. Now, I'm just going to show a couple of additional slides for a couple of different reasons, since we still have a little bit more time. And this was just a slide that I was working on with my team that kind of captures some of the opportunities for integrative multi-omic studies we have with these 2 collections, lupus and Sjogren's, which once again have overlapping disease features. For lupus, we have deep phenotyping, DNA sequence data, DNA methylation data, exposomics, metabolomics, data related to flares. On the Sjogren's side, through efforts that I haven't referred to this morning, in addition to DNA sequencing and DNA methylation, we also have single-cell RNA-seq and site-seq in peripheral blood, and we have DNA methylation data as well as single nuclei RNA-seq and bulk RNA-seq data generated in the gland tissue. So many opportunities for applying these multi-omics approaches to try to understand this heterogeneous disease. I wanted to take a minute to introduce members of my NHGRI lab. I am so privileged to be working with these 4 amazing people. Christina and Joanne moved with me from the beautiful Bay Area to the NIH, and Joanne now serves as my lab manager. Christina is a staff clinician. Mary joined our group as a postdoc, as I mentioned, and then very recently, Megan Nelson joined our group as a NIAMS translational scholar upon completion of her pediatric rheumatology fellowship at Emory. And it takes a village, as you all know, and highlighted here are some of our many important collaborators, including the increasing number of folks that we're collaborating with here. Thank you all so much, and many funding sources, and grateful, generous, committed patients. And I'm going to show you 2 more slides just to give you a picture of a couple of other things I'm working on in case they're of interest to those of you who I'm meeting with later. One is I'm going to mention that we were intrigued to see that this process of somatic mutation that's referred to as clonal hematopoiesis of indeterminate potential, which is the age-related acquisition of somatic mutations, 80% of those mutations occur in DNMT and TET genes that are directly involved in DNA methylation. So there's an important link there. And in that cohort of 2,000 individuals, we're characterizing them for these mutations in collaboration with Alexander Bick at Vanderbilt. We also, through that CLUES cohort, some states, such as California, have a program or initiated a program typically in the 80s where at the time of birth, parents are allowed to consent to collection of a newborn blood spot. We've been able to determine and get consent from 33 CLUES participants who were born in California when this program was in place. Their parents agreed, and they've now given us consent to go retrieve those blood spots along with match controls and look at methylation patterns at the time of birth before disease. So that'll be an amazing opportunity. And I feel so fortunate to have been able to receive an award from the Intramural Targeted Climate Change and Health Program. Thank you all for making that happen. And the focus of that grant is definitely on methylation and the methylome, but it actually focuses on the effects of wildfire smoke exposure on the epigenome and health. And this leverages a unique multi-ethnic cohort that was created to study COVID in the San Francisco Bay Area, but the timing of the biospecimen collection allowed us to collect samples before and after major wildfires swept through that area. So really excited about that work. And on that note, I'm going to stop, and I'm happy to take any questions at this time.
So for those in the room, please come to the microphone. And then for people tuning in online, please put your questions in the Q&A.
That was amazing. And thank you so much for just all of the things that you have shown us. It's very inspirational and you know, moving things forward into understanding such complex diseases. This may well be known, it's been a long time since I did rheumatology, but I'm really interested in the sex differences in, you know, in lupus in particular, and sort of wonder, I mean, do we know that that's sex steroids? Do we, and if so, in any of the studies that you've done, particularly the methylation studies, you know, have you cut that by sex?
Yeah, common question, and it's still something we're struggling to understand. So there's no question that hormonal factors play an important role, and there's variation in prevalence for different age groups, et cetera, et cetera. We also know that there are some genes on the X chromosome that have been strongly implicated in genetic studies. I've been fascinated by some of the studies of Klinefelters and Turner individuals, so those are folks that have either XXY or XO. Their risk of developing lupus and related autoimmune diseases reflects not their phenotypic appearance, but rather the number of X chromosomes they have. And then more recently, actually a NIAMS awardee published some very interesting work related to EXIST, which is critically important for X chromosome inactivation that occurs, you know, in women for one of the X chromosomes, and it turns out that that process elicits autoantibody production. So we think that probably the process of X chromosome inactivation increases risk for autoimmune disease, and we also know that escape from X chromosome inactivation contributes to autoimmune disease. The dosage on X. Now, a complicating factor for both genetic and epigenetic studies have been to try to fully analyze in a comparable way the X and Y chromosomes, and we're actually, Megan is going to take this on as a project that she works on to try to circle back to some data in which the X and Y chromosomes have been overlooked for kind of analytical complicating factors. So thanks for the question. I wish I could answer it fully, but there's a few comments. Hey, Rick.
Well, thanks very much, Lindsey. Is this on? Yeah. Thanks for a great talk. I've got a number of questions. I'll ask two of them now, and I'll see you a little bit later on. First of all, you have the advantage of being in a position where you have the genetics. You've got lots of different loci that have been implicated. And you're also looking at the epigenetics, the methylation sites. To what extent do these crosstalk with each other? So, for example, how often do you homozygose a locus by inheriting an allele, say a hypomorphic allele from a parent, and inheriting a complementary allele, an epigenetically modified allele that may be ratcheting down the level of expression?
Yeah, I don't know how to exactly answer your question, except what I can say is that obviously genetic variants don't fully explain disease risk, and there are many reasons for that. Part of it has to do with differences across different cell and tissue types in terms of how genes are expressed. DNA methylation controls that process to a great extent. But we also understand that in some cases, the way in which genetic variants influence disease risk is through differences in methylation. The cis-MEQTL analyses I referred to try to understand in which circumstances first of all, is it methylation pattern under genetic control and through causal inference testing is the methylation process mediating genetic association with disease. So when we do that, we see that absolutely those two things are integrally related in some cases, but in the majority they aren't. So in other words, DNA methylation is telling us something above and beyond what we can glean from the underlying genetic sequence. So there's a very partial response to your question, and maybe we can drill down
We
deeper
can talk some more.
during our meeting.
Another question is, are there any mouse models or any other animal models that may help you to work on this problem? It's a very complicated problem, whereas this kind of uniquely, you have to work in humans.
There absolutely are some mouse models that have helped us learn a lot about DNA methylation. In fact, the other slide I like to show, and I didn't today, was about the profound impact of methylation status on phenotypes. There's a Agouti mouse model, which has been studied primarily for its phenotype, which is obese, diabetic, and it also happens to have a yellow coat. And simply by giving those mice a diet high in methylation content, the phenotype is transformed into non-obese, non-diabetic brown coat, and that's a stable phenotype that can be maintained or reversed with a demethylating agent. So, like, wow, talk about a profound impact of that. So I'm not familiar, but there may be, with mouse models that are, kind of taking some of the emerging genome-wide methylation data and circling back and studying specific sites in animal models of autoimmunity. I would assume there probably are, but I'm not familiar. But I hope that by communicating to audiences like that, we can stimulate more basic research to try to understand what's going on here. What are the underlying mechanisms?
Thank you.
Please identify yourself at the mic.
Yeah, thanks, Lindsay. My name is Jeff Ginsberg. I'm with the All of Us Research Program, and it's great to have you here in North Carolina. I'm intrigued by the dynamic nature of methylation that you showed us with the individuals who underwent remission. So I have kind of two questions. One is, when you or others collect samples for exposomics, methylation, or multi-omics studies, do we need to know the timing of that sampling from the last flare so we can potentially control for that variation that you showed us? And the second question is, do you think there's an opportunity for looking at those that achieve remission to do GWAS and those across those that have remission versus those that don't, to look for effectively resilience loci?
Yeah, I mean, so it was very interesting the extent to which, and it's a dynamic process. It was the changes in methylation patterns that were strongly associated with remission or not. It would be very interesting to take those subsets and look at them from a genetic analysis standpoint. So to what extent would that have been predicted by genetic factors? In terms of the timing, yes, we really need longitudinal data and biospecimen collection. One of the things, I was mentioning this in one of my meetings yesterday, I think, we have tried, and I've alluded to it, whenever we collect a biospecimen, what meds are they on? All of these things. It's not always possible to get a disease activity measure because it's like 59, questions. It's complicated, but whenever possible, we try to understand that, to adjust for that or to stratify by that. But what I'm really excited, and I'm hoping I can establish collaborations at NIAMS or elsewhere to do this, is to look at clinical trial populations and collect samples at baseline, during the trial, following the trial. Because when we try to adjust for medications in the context of observational data, it's really challenging. We published a relatively small rheumatoid arthritis study last year in which we looked at methylation difference changes among rheumatoid arthritis patients who were treated, a new start on methotrexate, and based on their responses. And it took us 5 years to recruit the requisite number of participants to kind of do the study. And if we were embedded within a clinical trials program, we could do that so much more efficiently. We wouldn't have to worry about all of the, some of the covariates that we're not capturing. So, yeah, thank you.
Thank you.
Hi, I enjoyed your talk. I'm Franco DeMeo, Chief of Reproductive and Development Biology Lab, and I'm not going to ask a sex story question. So you're doing a lot of, the sites you're looking at, their susceptibility locus because of the variability of the disease, right? So there must be something tweaking those. So you're going to do single-cell RNA-seq. Do you think it'd be good to include open chromatin assays like single-cell ATAC-seq and then try to come up with what are the central regulators of this and what maybe the triggers for the variability in the disease?
Absolutely. In fact, I didn't mention one of the collaborations that I'm really excited about is going to be with the NIAMS Genomics Technology Core, where in the clues cohort, the FLAIR samples, we're going to generate single-cell RNA-seq, ATAC-seq, methylation data, et cetera, same individuals, same samples, et cetera. So we're really excited about, that is, as good as you can get in terms of an environment to try to understand how these things are interrelated. But, yeah, Daryl.
Yeah, so Daryl Zeldin, Scientific Director. So great talk. And it's wonderful that you're doing this multi-omic approach to rare but very significant diseases. So my question is more about how what you're doing is going to ultimately influence therapeutics and particularly personalized medicine. So, I mean, knowing the multi-ohm in these patients, so how is that changing, or how will that change over the next 10 years, how we treat patients with lupus or Sjogren's? And how can we take a personalized medicine approach to the information that you'll glean on a patient-by-patient basis and use that to decide which are the best treatments in a particular population?
Yeah, I mean, a lot of pieces to that. One thing I will say is as I mentioned, there are a lot of problems with the classification criteria. Not only do they encompass a lot of heterogeneity, but they're not driven by some biological insight or understanding of the disease. So they're just based on observation. So we know there is heterogeneity within those criteria, and we think that's probably one of the reasons why most clinical trials in lupus have failed. We don't know how to select the right participants for the right drug. So first of all, even if we don't understand the underlying biology, if we can, through some combination or some subset of genetic, methylation, other multi-omic data, identify subsets of disease that seem to share pathways or are distinct from the rest of the lupus patients, and then start applying those classification schemes to clinical trials, I think our progress will really evolve. But also, it's not difficult to imagine that as we identify pathways that are operating in certain subsets of patients, it will suggest new therapies. I was really excited. This is a very old study now, but as GWAS data was emerging for rheumatoid arthritis, this talented fellow took the emerging GWAS data and just mapped it onto drug databases and what drugs are already approved, being used in other indications that might be appropriate for this rheumatic disease. And there were 2 or 3 therapeutics already approved in cancer that could be quickly, drug repurposing. I think that holds tremendous promise. So I think that sort of approach. Can I point to a specific example where we've said with this data or this combination of data, boom, we now know how to treat this subset? No, but I think we have tools that will allow us to get there. And by the way, one last thing, the vision is not that when patients come into the clinic, we're going to have to get 10 tubes of blood and do all of these multi-omic analyses. But by having multiple layers of data and then analyzing them through these data reduction approaches. I'm excited by this multi-omics factor analysis approach. If we can figure out which layers of data or if we can simplify, take all of this data and understand what are the essential factors that are influencing the disease, then we might have something that could be applied at the clinic. But I'm hoping that we're looking in the next decade at revising all of these classification criteria based on molecular data. Fred.
Great talk, Lindsey, and it was a great summary of a lot of complex and difficult to obtain data, I know. And not too many people in this audience appreciate the complexity of getting those cohorts together and figuring that all out. It's Fred Miller from the Environmental Autoimmunity Group. I am impressed by all the data that you presented, but of course the environmental side is still the challenging aspect of this. And I know you have some data already, smoking and a lot of other environmental information that's been collected. The medications you talked about briefly, I know the challenges there, but at least the medications at the time of the collection of the DNA might be useful. Are there ways that you're thinking about trying to put all these environmental exposures together in a interesting gene-environment interaction approach to these diseases, which is where I think the key to all of our questions really lie?
Yeah, I mean, since I was a little kid, I've been fascinated by this nature versus nurture genes and environment piece. And, the environmental piece has been much more complicated, but we're getting there. So Absolutely. I mean, we have polygenic risk scores. We have polygenetic methylation scores. So how about poly-environmental, risk factors? I think that, and I think the ideal is complementing this sort of profiling approach with rigorous epidemiologic study designs. But I hope we can, imagine a future where we understand the major environmental contributions to disease. And by the way, we still don't know when during the course of a lifetime is the critical time point. Is it the in utero exposures, et cetera? So I'm not sure how we're going to get there, but I can imagine a time when someone comes into the clinic, maybe they have relatives with systemic lupus or they have early symptoms, and we assess a dozen exposures that we feel convinced based on anecdotal, epidemiologic, MS data are important, and try to counsel them or just try to understand their risk of disease. Smokers, for example, people with a significant smoking history are much more likely to have severe rheumatoid arthritis, to have lupus characterized by double-stranded DNA and autoantibodies, and it's helpful to understand and to be able to project what their disease trajectory is going to look like, how aggressive to use DMARDs or biologic agents, et cetera. So we'll get there.
Thanks a lot. Those are all great ideas.
Yeah, thanks.
Okay, we might take one of the online questions before we end. If there's been any studies examining transgender or intersex individuals with these diseases, particularly in lupus?
Yeah, that's a great question, and not that I'm aware of, but does anybody, I know you have a big reproductive endocrinology presence here. Does anybody know of a resource for investigations in that really an interesting population? Yeah, there was a fellow here who promised not to ask any sex steroid questions. I'll have to catch him later. So not that I know of, but wow, it'd be really interesting to see what's happening in terms of development of disease and progression of disease among some of those individuals.
Okay, well, we're at the top of the hour, so I'd just like to thank you so much for your wonderful presentation today, and we're again looking forward to your visit here today at the Institute, and thank you again.
Thank you so much for the honor. Really appreciated it.